So I'd like to ask you just for a moment to imagine a situation is a child who all of a sudden can't express words and lost their ability to speak and to connect. And that happened because of hidden changes in their RNA. So today I will walk you through how a variant, an ultra rare variant in the gene HNRNPH2, which I will call H2 just to make it easy, can change RNA regulation in a human brain organoid model with a special focus on splicing. So over the next few minutes, I will guide you through organoid model validation and global differences in the transcriptome and different layers of splicing to finish with an integrated view that highlights some key regulatory hubs. Mutations in H2 cause an X-linked neurodevelopmental disorder that leads to serious language and motor problems and often autism. The R114W on the H2 gene is ultra rare, and it happens in a part of the protein that binds RNA. And this is why we focus on RNA-based outcomes. We start with a patient-derived iPSCs carrying the R114W mutation, what we call the minus line, and compare them with a CRISPR-corrected control, or plus line. These two are genetically identical, except for the R114W change. Both lines go through the same cerebral organoid protocol, so any differences we see are related to the genotype and not to the background. And first we check that the starting IPs are similar. So both minus and plus lines show the same pluripotency markers, both 5 and anog. We also look at H2 and SHANK1 to confirm that we have a matched baseline. And this tells us that pluripotency is preserved in both genotypes. So any later differences are not caused by unequal starting cell states. As the cells differentiate, we then track their stage-specific shapes to make sure the process is consistent. In mature organoids, we use key markers to check the identity of these new cells. For an example, PAC6 will mark cortical progenitors, while TUB3 labels mature neurons. And then we compare these markers between the two genotypes, and we see that both minus and plus organoids reach the expected functional endpoint, so differentiation works in both cases. After confirming maturation, we check a few more phenotype panels for stress pigmentation, synaptic markers. We measure G3BP1, tyrosinase, SHANK1, and H2, and see that both genotypes express these markers, though some profiles differ between minus and plus. And these divergent profiles raise a key questions. Could this early hint signal deeper disruptions in RNA processing, such as splicing shifts? And to address that, we move to a transcriptome-wide approach. Here we show how our samples relate to one another using a sample similarity heat map and dimensionality reduction. When we ran PCA to compare minus and plus at the whole transcriptome level, the two genotypes form clear separate clusters. Aligned with this, the volcano plot translates the global separation into gene-level changes, revealing which transcripts shift most strongly between minus and plus. And the broad spread of significant points in the volcano indicates that the genotype effect is widespread at the gene expression level, rather than just driven by a few outliers. And this separation encourages us to examine some specific RNA layers in more detail. So to understand transcriptomic changes, we first present gene-level differences using volcanoes and heat maps. But instead of looking at single genes, we use enrichment analysis across gene ontology, KEGG, reactome, and related pathway sets to see which programs change together. The most enriched terms include neurodevelopmental programs, among genes downregulated in minus, like neuron differentiation, axon development, synaptic transmission. And on the other side, signaling and metabolic programs among genes upregulated in minus, including Wnt and NOT signaling, and also pigment and metabolic pathways like melanogenesis, tyrosine metabolism, and retinoic acid biosynthesis. So for an example, an overrepresentation analysis can show that axon development terms are overrepresented within the downregulated gene set, while melanogenesis-related terms are overrepresented within the upregulated gene set in the mutant organoid. And at this point, we connect our findings to a possible mechanism using structural modeling. But before we look at the docking model, it's important to ask, what might this mean for RNA-based therapies? So if the mutation alters the way that H2 interacts within the spliceosome, can it really open the door to targeted therapies that restore proper splicing? Our docking model shows that H2 within the spliceosome and highlights residue 114 at an RNA binding site, supporting the idea that the mutation disrupts RNA binding and could cause widespread splicing changes. So now for the main result, we study splicing from both the junction and axon perspectives. And to make this contrast more intuitive, just think of axons as entire city blocks and junctions as intersections where roads meet. So if you only look at how each block is used, you might miss major traffic jams or detours that happen at the intersection. So precisely, these are the places where things go wrong in disease. So our heat maps and volcanoes show that junction uses is the clearest difference between minus and plus, while exon-based signals only tell a small part of the story. And we use overlap panels to show which genes have consistent splicing signals across methods, and volcano plots to compare effect sizes and significances. So what we see is that splicing problems are mainly driven by what happens at the junctions, while exon-based views miss much of the signal, just as focusing on blocks would overlook critical problems happening at the intersections. So the truth is that results from a single layer can be considered fragile, and we decided to rank genes by combining evidence from gene expression, from splicing events, and isoform changes. So we have a coupling strength heat map to show integration and highlight candidates supported by several metrics. And we focus on minus versus plus comparing and highlighting a coupling geometry where we test the relationship between events and isoforms, a dominance map where dominant genes on both event and isoform changes, which means if there is more modifications happening at the junctions to the point of disturbing the whole isoform. And we also plotted a signal geometry to compare the transcriptional amplitude versus RNA disruption. So until which point these modifications in splicing can really disturb the expression level of a certain gene. And finally, we asked if cell type composition could explain our results. So we decided to use MUSIC, which is a deconvolution package based on single-cell-seq. And we analyzed these samples with a neurodevelopmental reference based on single-cell sequencing. And we estimate cell type fractions and check the stability to compare these shifts and to mark gene patterns. And while cell type differences are real and reproducible, the magnitude is modest. So across samples, neuronal fractions in minus organoids are about 5% lower on average than in plus organoids. And that supports the conclusion that cell composition is just a secondary factor and not the main driver of genotype-related changes in the RNA landscape. So looking ahead for the next steps, we hypothesize that antisense oligonucleotides can restore normal splice junction usage and rescue key pathways disturbed by H2 mutation. And to test this, our next step is to use the junction and isoform level changes as a clear readout for tracking how well a therapy would work. And if this hypothesis is correct, these results may redefine how we approach treatment for neurodevelopmental disorders. So on the left side, we can see a spatial mass spectrometry image showing that one of the ASOs tested passively diffuses the organoid membranes, reaching the inner core. And on the right, a preliminary Pearson correlation indicating that we are on the right track. So I just think that we should imagine a future where a child once struggled in silence because of an RNA mutation, and then she would be able to speak and to reconnect again. And that's the kind of possibility driving our work forward. And I'd like to thank you. Thank you.
Yes, many thanks, Carlo. Trying to activate also my camera. How? Yes. Yes. Okay. Many thanks. Yes, I think there is yeah, for your work, and like this really it seems like you found like a bit of a non-canonical function for this protein, because I read a little bit and it seemed to be like responsible for the transfer to the nucleus, to the transfer of the RNAs in the nucleus. And this seems to be also another function in the splicing, which makes it very interesting, and then a different mechanism. And I also know a struggle of working with the rare diseases, and potentially, yes, like it's wonderful how you can model it in the stem cells now. So while we maybe have time for one question, but we're still waiting for the question to pop up in the chat. Please write the questions, and maybe I can quickly ask if you know if this mutation is like a germline, or is it somatic? Is it happening at some point during neurodevelopment, or is it present already at the very start?
So, yeah, we suspect the mutation is more active in some types of cells than others, right? So mostly, and I would say, I think it's important that H2, HNRNP, H2 gene is not really well characterized. Like 5 years ago, we had less than 20 articles about this gene.
Yeah.
And now we had 5 years from now 26, we might have around 50 articles for this gene, right? And most of the information came from autistic patients. And there are two main points in the gene where these mutations can cause autism. Most of them, right, more than 10 patients, presented mutations at the nuclear localization signal. And this is why there's a lot of focus on H2 function as a protein that will enter the nucleus because it has the NLS, right? But as the gene I'm studying, the mutation, the variant, ultra rare variant we studied, came from a patient. I met this patient, and only 2 patients in the world were recognized with this mutation. And it happens in the RNA recognition motif, which close to the NLS, but not in the same area. And so this protein has, this variant, this gene has the perfect NLS, so it can enter the nucleus.
Yes, okay.
Splicing is altered, right? And then we began thinking that an antisense oligonucleotide therapy would be able, like a gapmer ASO, would be able to knock down H2, and we have another isoform in the human genome, which is HNRNPH1, which 98% close, would restore the function. So we then developed ASOs to study how an ASO can really rescue this cell's phenotype and probably be used as a therapy.
Yes. No, this is definitely exciting. Wishing you all good luck with the further experiments.